My name is Julia Stockham and I work at Solinta as an agronomy and physiology researcher. Solinta is a breeding company for diploid hybrid potato. We produce homozygous inbreeding lines by performing several generations of self-pollinations. By crossing two inbreeding lines, we produce true hybrid seeds. Advantages of using true seeds instead of seed tubers as starting material are, for example, the easier and cheaper transport and the possibility of fast upscaling of seed production. Besides that, true seeds are clean and disease-free, while in seed tubers, diseases easily accumulate. Also for breeding and research, there are advantages of this diploid breeding system. Genetics are less complex in diploids than in tetraploids, and the possibility to produce homozygous inbreeding lines and segregating populations make genetic research more efficient. When breeding new varieties, the selection of desired traits and marker-assisted backcross breeding becomes faster and easier. In this project, we want to make use of the advantages of the hybrid breeding program to produce resource-efficient varieties. Together with Xavier Draay from the University of Louvain, we are part of the Horizon 2020 project Soleis, in which we aim to grow nutrient and drought-tolerant plants. In that project, we performed some greenhouse and field trials under control and stress conditions. However, in these trials, it was out of the scope of Soleis to perform trials in which we could investigate specific root traits. Xavier discussed with us the possibilities of the aeroponics facility in Louvain, and that this would be a great opportunity to measure root characteristics of potato seedlings. We applied for this EPPN project to be able to identify functional root traits in potato seedlings that improve growth under nitrogen-deficient conditions. For this purpose, we selected a panel of 60 contrasting hybrids, which you can see in the video right now. The parents of these hybrids were genotyped, and this information will be used to map the root traits that were found in the screening. This was the first time that hybrid potato from true seeds were grown in the aeroponics facility. Xavier and his team therefore first performed several pilot trials. The challenge was that the roots of the seedlings were very thin, they stuck together and could not be measured. The solution was to make cuttings of the seedlings instead of using the just germinated seedlings for phenotyping. Therefore, for the actual trial, we decided to grow seedlings and make cuttings of the seedlings that were later transplanted to the phenotyping facility. Here you see the seedlings that were grown for the trial when they were about 3 weeks old. Here you see how the potato plants were growing in the facility and how the roots grew in the aeroponic system. The root development can be followed in detail without damaging the plants or roots. Traits like root length, elongation rate and lateral root density can be determined. Roots were screened in 2-hour intervals like is visible here. As such, differences in root growth and architecture between treatments and genotypes could be measured in time. We found variation in root length and in lateral root growth between the genotypes as well as between the treatments. Next steps will be to map functional root traits and to make them available for breeding.